Hello guys, welcome to Amazon Academy. It's a wonderful platform to prepare for different competitive exams like IIT JEE, NEET and CUET. So guys, today we are going to discuss one more factor which can affect the enzyme activity, that is inhibitors. Okay, this topic is taken from class 11th biology, chapter name is biomolecules. Okay, so let's get started. Okay, so first of all, let's again let me brief you with certain basic points about enzymes. That suppose this is the enzyme. Okay, these enzymes have these active sites. Okay, what are active sites? Active sites are the sites of protein which are in their three-dimensional structure. Okay, tertiary structure. Okay, which binds with the substrate. Okay, and these active sites are specific for their enzymes. Okay, so active sites are specific for their enzymes, for their substrate. Okay, why we are reading about active sites? Because this is going to come when we talk about inhibition. Okay, so active sites are specific for the substrates. Okay, suppose we have a substrate like this. Okay, like this. Okay, and this is one more substrate. Okay, different substrate. Okay, so we have enzyme which have active site corresponding to suppose this is substrate A and this is substrate B. Okay, so we have this enzyme, okay, which has active site corresponding to this substrate. Okay, like this. So this substrate can bind here, okay, like this, and form the enzyme substrate complex. Okay, so in this way every enzyme have active site which is specific for a certain kind of substrate. Okay, so what are inhibitors then? Inhibitors are the substances which when bind to enzyme prevent the catalytic activity of the enzyme. Okay, so let me define. Okay, what are inhibitors? Inhibitors are the chemicals, okay, which on binding with the enzyme, okay, with the enzyme, okay, can stop catalytic activity of the enzyme. Okay, of the enzyme, that is they will stop the enzyme function. What is the function of enzyme? That they increase the rate of reaction by decreasing the activation energy. Okay, so chemicals which can stop this are known as inhibitors, and the process is known as inhibition. Okay, process is known as the inhibition, or we can simply call it enzyme inhibition. Okay, enzyme inhibition. Okay, enzyme inhibition. Okay guys, now as we discussed in the previous classes that we have different factors which can affect enzyme activities, such as temperature. This we have discussed already. pH, effect of substrate concentration, okay, and finally our inhibitors. Okay, inhibitor. So in this session we are going to talk about mainly about inhibitions. Okay, so enzyme activity, effect of inhibitors. Okay, so enzyme inhibition. Okay, it can be of various type, like it can be reversible, it can be irreversible, okay, or it can be competitive, or it can be incompetitive. Okay, we are mainly focusing on competitive and incompetitive inhibition. Okay, competitive and non-competitive inhibition. Okay, non-competitive inhibition. Okay, so let's start with discussion about competitive inhibition. Okay, so first, what is competitive inhibition? You can take example from yourself only. There is one seat in the medical college and number of students are competing with each other for that MBBS seat. Okay, so same thing is going to apply here. So competitive inhibition. Okay, so suppose we have this enzyme. Okay, suppose we have this enzyme and this is the active site. Okay, this is the active site, this is the enzyme, okay, and this is the active site, okay. Now this enzyme normally binds with the substrate, okay. So this is the substrate, suppose, okay, they normally bind with the substrate and our product is formed. But when a similar looking chemical, okay, which we'll call it as the inhibitor, okay, suppose we have this similar looking which can also bind with the active sites of this enzyme, okay, is there, okay. So since both of them can bind with the active site, they both compete with each other, okay, for the active site of the enzyme. So in this, what is going to happen, that both substrate and inhibitor are going to compete with each other for active site of enzyme, okay, for active site of the enzyme, okay. Now why it's possible? Because inhibitor resemble substrate structurally, okay. So inhibitor resembles substrate structurally and can also bind with the active site of the enzyme, okay. Since they both resemble each other, okay, that's why inhibitor can also bind with the active site. Now in normal condition only substrate bind with the active site because substrate is specific for the active site, but since inhibitor resembles the substrate structurally, so inhibitor can also bind with the active site, okay. So since inhibitor resemble the substrate structurally, so inhibitor can also bind with the active site. Now on which factor it will depend, that whether substrate will bind or inhibitor will bind, it will depend on their concentration, okay. So we can take two situations here. Suppose we have substrate is more than the inhibitor, okay. So possibility of, or you can say probability of binding of the substrate with the active site is more than the inhibitor, okay. Suppose we have this container, okay, and we have the enzymes here and they have the active site like this, okay, and we have new number of enzymes, okay, and then we have the substrate molecules. Suppose these are the substrate molecules, okay, they normally bind with the enzyme, okay. So these are the substrate molecule. But when we have similar looking inhibitor molecules also, suppose these are the inhibitor molecule, okay. So it only a interplay of the concentration. So if substrate molecules are more in concentration, so there will be more probability of substrate binding with the active site. So there will be no inhibition, no significant inhibition, we can say no significant inhibition, okay. But when it is in reverse condition, that is we have more of inhibitor as compared to substrate, okay. So again I'm going to draw the same vessel, okay, and we have these enzymes like this, okay. So these are the enzymes and now we have more of inhibitors as compared to the substrate. Suppose these are the inhibitors, okay. So and less of the substrate, okay. Suppose these are the substrate and they are less in concentration, so these are substrate, okay. So now when inhibitors are more, so probability of inhibitor binding with substrate increases, okay. So in second case what is going to happen, suppose this is the enzyme, okay, this is the enzyme, okay, and this is our normal substrate, okay, this is the normal substrate, okay. So normally substrate bind with the enzyme and our product is formed. But when we have the inhibitor, okay, suppose this one which is more in concentration than the substrate, okay. So when inhibitor is more in concentration than the substrate, so in that condition what is going to happen, that enzyme is going to bind with the inhibitor instead of the substrate. So this is the enzyme, this is the active site, okay, this is our active site, okay, like this. Again, this is the active site, like this, okay. So in this condition, what is going to happen? That substrate is not able to bind because substrate is less in concentration. So now inhibitor is going to bind, okay. And when inhibitor is going to bind, there will be no product formation because product is formed by the substrate, not by the inhibitor, okay. So this is the inhibitor, okay. So since substrate is unable to bind with enzyme, okay, so therefore no product is formed, okay, no product is formed, okay guys. So let's take one example, okay. I think you have read about Krebs cycle, okay. So this example is taken from Krebs cycle, okay. We have a substrate, okay. Substrate is known as succinate, okay, succinate, which is converted to fumarate. Fumarate is the product, okay, by an enzyme known as succinate dehydrogenase, okay. This is the normal reaction, okay. This is the enzyme, okay. The substrate combines with the enzyme succinate dehydrogenase is converted into product, okay. But in case of competitive inhibition, now why it is called as competitive inhibition? Because the substrate and our inhibitor are competing for the active site of the enzyme, okay. So what is called as competitive inhibition? Because substrate and inhibitor are competing with each other for active site of the enzyme, okay. So in this case the inhibitor will be our malonate, okay, malonate, okay. So instead of succinate, malonate is going to bind with the enzyme, okay. So this malonate, it binds, this is the inhibitor, it binds with the enzyme, okay. Enzyme is our succinate dehydrogenase, okay. This is the enzyme, okay. So when inhibitor binds with the enzyme instead of substrate, so there will be no product formation, okay, no product formation. So we can say activity of enzyme is inhibited by the competitive inhibition. So I hope this example is absolutely clear in your mind. This example has been asked number of times. So you have to remember the inhibitor, which is the inhibitor, which is the substrate, which is the product, and what is the enzyme, okay. So I hope this concept is clear in your mind, okay. So let's jump to the next type of inhibition, okay. Second is non-competitive. Again, we can take examples. Suppose you are preparing for NEET exam and your friend is preparing for IIT exam. So there is no competition amongst themselves, okay. So no non-competitive inhibition, okay. So what is going to happen here, that the inhibitor is going to bind to some other site than the active site. So inhibitor is going to bind on site other than the active site, So what is the problem? Then inhibitor is binding to a site which is not the active site. So what is the problem? The problem is that when inhibitor binds with the site other than active site, it leads to configurational change in protein structure, okay. Protein means enzyme here, okay, which leads to change in the structure of active site, okay, change in structure of active site, okay. So now substrate is unable to bind with the active site because there has been change in the structure of the active site, okay. So substrate is unable to bind with the active site, and since substrate is not able to bind with the active site, so there will be no product formed, okay. There will be no product formed. So we can say that enzyme is inhibited. Okay, enzyme is inhibited. Okay, let me show you or explain you with the help of a diagram. Okay, so let's move to the next page. Suppose this is the enzyme. Okay, this is our enzyme. Okay, okay, so this is the active site. Okay, this is the active site. Okay, so this binds with the substrate. Suppose this is the enzyme. Okay, and it binds normally with the substrate. Okay, this is the substrate. Okay, something like this. This is the substrate. Okay, now we have an inhibitor. Okay, this inhibitor, suppose this is the inhibitor. Okay, this inhibitor does not binds with the active site. Okay, it binds here. Okay, let me draw it again. It binds here, which is a site which is not the active site. Okay, so when this binds here, okay, suppose this is the enzyme. Okay, let me draw it again. Okay, this is the enzyme. Okay, like this. Okay, so when this binds here, the inhibitor, it is going to cause change in the configuration of protein, including the active site. Okay, so there will be change in the shape of the active site. Okay, so the active site now becomes something like this. Okay, like this. Okay, so you can see there will be change in the active site. Okay, so when this is the inhibitor. Okay, so when inhibitor binds with site other than active site, what is going to happen? There will be change in shape of the protein and also of the active site. Okay, we can simply write active site. Okay, so now we have this substrate. The substrate will going to remain the same. So now as you can see, the shape of the active site has been changed and now it can no longer bind. Okay, so substrate is unable to bind with active site. Okay, unable to bind with the active site. Okay, so no product is formed. Okay, no product is formed. So this is the non-competitive inhibition. Okay, so to put in a simple words, in competitive inhibition, both inhibitors and substrate are structurally similar. So both of them can bind with the active site. It only depends on the concentration. If substrate is more, probability of binding the substrate is more with the enzyme. If inhibitors is more in concentration, probability of binding or colliding of molecules of inhibitors with the enzyme is more. So in that case, if inhibitor binds with the active site, there is no formation of product. Okay, in non-competitive inhibition, there is no competition. Inhibitor is going to bind to some other site like this, but when it binds with some other site, this is going to cause change in the shape of the active site. So now active site is no longer able to bind with the substrate and there will be no product formation. Okay, let's take an example. Okay, example of this kind of inhibition is inhibition of cytochrome oxidase, which is a very important enzyme in respiration, by cyanide. Okay, so when cyanide binds with the cytochrome oxidase, it can no longer bind with its normal substrate and there will be disruption in the respiration mechanism and patient is going to die because there will be no substrate, no product formation. Okay, now what is the significance of this kind of inhibition? Significance of inhibition, okay, that these all the drugs we take, like antibiotics in normal drug, all the drugs we take, they are basically inhibitors of some enzymes. Okay, inhibitors of some enzyme of our body. Okay, so I hope now your concepts are clear. Thank you very much for your time. Till next time, take care and bye bye. Okay, and happy learning from the Amazon Academy. Okay guys, take care and bye.